right, moving on. Here we are in section 10.3. Now we're going to be looking at the actual production of sugars through the Calvin cycle. So, the Calvin cycle, spelled something like Calvin, is an anabolic process occurring within the stroma of the chloroplast. So in that thick viscous fluid, we're going to be making some sugars. Now, the basic concept is we are taking carbon dioxide and reducing that carbon dioxide to create organic molecules or sugars. Now, to do this conversion, it's going to require energy. The energy is going to come from ATP and the reducing power of NADPH, two molecules formed during the light reaction. So the Calvin cycle is called the light-independent reactions, the dark cycle sometimes, but that's only because it doesn't directly require light. It does indirectly require the energy from light, just in the form of these molecules that were made earlier. Now, I'd like to first look at the Calvin cycle visually. I'm having some serious Calvin spelling problems here. We'll just call it the Cal cycle. The Calvin cycle begins with CO2 that is then joined up with a molecule called RUBP. Not a question, a molecule. So here we have RUBP combining with carbon dioxide with the help of an enzyme called rubisco. Rubisco helps join CO2 to this carbon carrier RUBP. Now, this creates a molecule called 3-phosphoglycerate. Now, I don't anticipate asking this on any problems on a test or quiz, but I just want to put it out there that RUBP joins with carbon dioxide with the help of rubisco to form this brand new molecule. Now, in this process, ATP is used and NADPH is used forming NADP plus and hydrogen ion. So these molecules are used to generate a molecule of G3P. Now this is the molecule that will actually split out and become the sugar. So glucose doesn't get spit directly out of the Calvin cycle. Hey, spelled it. Glucose isn't spit out of the Calvin cycle directly. It's this G3P molecule, and then that can combine with other 3-carbon molecules to form glucose. You can form sucrose. The glucose monomers can join to form starch. Any assortment of organic molecules can be made. Now, what will continue to happen is some of the GP3 molecules will continue on through the cycle, and again through the use of ATP get reorganized back into RUBP. So that is why this is referred to as a cycle. So if we think about it here, RUBP is combined with carbon dioxide using rubisco to form this molecule. This molecule then undergoes a series of reactions using energy and electrons from ATP and NADPH to form G3P, which will get spit out and converted into a sugar. Now the remaining carbon molecules, carbon-based molecules, are then rearranged using ATP yet again to form that original carbon acceptor, RUBP. Let's even give it that name. Okay. thus repeating the cycle. So as long as carbon dioxide is available, and as long as ATP and NADPH are available, you'll have this cycle continue with the continued production of these sugars, again, occurring within the stroma of the cell. All right, what I'd like to do now is look at this a little differently here. If I can get the thing to work. There we go. Sorry about that. I feel so adept with technology when I have to dial a phone. But beyond that, I'm always still a little lost. All right. Now, let's look at the Calvin cycle a little differently. Let's put it into sort of a flow here. Melvin Calvin would be upset if he saw how much I struggled spelling his name. All right, the Calvin cycle can be broken into 3 basic phases. You have you have the carbon fixation stage. So phase 1 is carbon fixation. Now, during carbon fixation, again, carbon dioxide is joined to RUBP. Again, RUBP is that carbon dioxide acceptor. And then this is done using the enzyme Rubisco, potentially the most prevalent enzyme on the face of the earth. Okay, and in doing so, in joining carbon dioxide to RUBP, you end up creating this intermediary molecule that will get passed on and eventually converted into sugars. Now, the second step is that sugar production phase. So step 2 is a set of reduction reactions. So in reduction, NADPH and ATP are used to make that sugar G3P. That is the sugar or the product that will eventually get converted into glucose, sucrose, starch, whatever the cell or whatever the plant needs. Okay, the third set of events is the regeneration of that initial carbon dioxide acceptor, or the carbon acceptor, which was RUBP. So we're going to focus on the regeneration of RUBP. Now, when this is done using power from ATP, this molecule can now fix more carbon dioxide from the atmosphere. So the cycle continues. So you can take what we cover here and look at the diagram that you just completed and see how the Calvin cycle basically is comprised of 3 individual steps.